New admission? પેલા હે હા ના એવું બને ને પેલી વાર દેખા થોડું સમજાવતા રહેજો શું કરું. Just good morning to all. So last we were talking about selective methods, right? In that previously we have seen the liquid and the characteristics particularly. When we are having the liquid process or when we are having the solid medium, either it is in slant or in a plates, we are going to observe their basic phenomena. What are their characteristics are there? That was our basic target. This point I have taken specifically selective method of isolating pure culture. It's in syllabus they have given in the first line, but I'm taking at the last. The reason behind it, when you have learned few concepts, then it is become easier for you to understand this part. Otherwise, it is going in air. So I thought to take at a last point into it. Okay. So what is the basic part over here? So microbial populations, when you are looking to, the students has just gone to in a lab to bring our plates. Just I'm going to show it that part. Okay. That is related to previously you have inoculated. But due course of time, I was also not available just yesterday. And work was on pending to just observe, right? So we are just going to check today that whether there is, you have done the inoculation, right, on the nutrient flex. You have just done the streaking part. Okay, that was the radial streaking you have done. There is a chances maybe your plates got contaminated either, or it may be having the pure colons. So it is just a basic selective method that you have done the streaking. But when you have the like natural microbial population is there, maybe you have taken the soil sample, or one person is very much sick, having the some digestive problem, diarrhea or something is there. We don't know. There may be thousands of reasons behind the diarrhea. Sometimes just something happens. Some person is get frightened up, right? They are telling that got diarrhea, right? Anything has happened in any conditions, then the everything is loosen up. There is no any role of any bacteria. No, nothing is there. It's a physiological change in our body, right? Other than that, there is a one particular situation and condition that microbial populations will be there. I have taken some few examples related to it only. That some bacterial populations are causing the diarrhea, and we want to check it out that whether this organism is there in a stool or not. It is really difficult because they are having some particular characteristic. It is our stool do have particularly like normal flora into it. So when you are going to take it out the some sample from it and going to culture it, our normal flora will grow fluidly. So we need to control them. It must be get controlled. Then only it's easier for us. So we need to go some selective process into it. Okay, otherwise it will be very difficult. So we need to have the selective methods. So we do have the physical, chemical, and biological, three different methods by which we may go for the selective particular kind of a bacteria. Okay, so getting the pure culture. So it is our basic desire. Okay, why we are looking for the selective culture or pure culture into it? That a desired beneficial or harmful organism we are looking to get isolated out of given the sample. It may be a soil sample. It could be a hospital-based any sample is there, or it may be a randomly water sample, or the sewage sample is also possible. Sometimes if you are looking for isolating the petroleum degrading, if you are targeted or the dye degrading, here we are knowing the west, you are looking and moving outside, like in Jaipur, and some people are going. Textile industries are there. Some other pharma industries are there. Some other pesticide, agricultural pesticides are there. A fertilizer company is there. So they are rich in some particular kind of a nutritive materials or the chemical compound. So what's going to happen? That some bacteria are start to grow over there. They will take it as their particularly nutritive material. They will digest it and they will grow. So what we can do, from same place, we may isolate some water sample. We may take the water sample. You may go in the sea also. Okay, that is a marine water. What you will get from the marine water? Marine water will be Salty, yes. So salt, that is a salt degrade, mainly the salt utilizer. We are not going to say as a degrader, salt utilizer. So halophiles, yeah, you can keep here. Yes, very good. After a week, we are going to have the lots of contaminations even the good plates. I think we have kept the plates with certain other objects, maybe a reason. Yes, no growth, but very easy. There is a no contamination. Okay, you can just pass it out. You may pass the plates. There is a mixer into it. Okay, you may take and just take it out. It's a mixer. Just try to locate what sort of the different. Don't open it because it may have the fungal spore. I'm going to say a few things that is like a dark. Okay, dark spots are there. Some are the yellow or the filamentous. Every sort of the thing is there. Very good. You got people lots of things. You can just pass it out. It is random. It is maybe your name is there or not a problem. But don't open. Just few things are there into it also. Some are very much blank. That is good. They have done maybe a now the maybe a two reason. We have kept within our incubator. It do have the fungal or the other spores are prevailing from the beginning. So is it one. Good. Let's. Yes. This one is particularly, if I'm not wrong, it may be Aspergillus niger. There is a, we are telling that a niger, there may be a not that much potential, but very much good source related to infections on a skin. Aspergillus niger, it is a similar, generally, where we are getting on the onions. On onions, sometimes you may find some dark spots are there. There are actually and the kala dots it is fully the Aspergillus niger. This one is good source of antibiotics even. If you want to do some research here, yeah. So these are the good source of antibiotics. Penicillin and others are also well known. We may go for chromics. So it may cause the infections even. Skin infections may be a possible. So there may be a chance. Earlier we have kept within the one incubator. It do have some raw materials. Fungal spore were there. They were looking to culture the fungal spore. So from the air, fungal spore may have introduced and contaminated your plates. So whenever you are going to keep for at least 1 week, 10 days, it should not get any contamination. Several plates are there. It doesn't have any sort of the things. Like Rajesh, written as Rajesh, maybe he has done properly. There is a no contamination. Okay, it is good sign. So there may be two things. While opening or handling, you may have were talking with each other. Okay, it is a routine. Okay, or else even the talking as well as you are not able to open properly. Some people are just helping you out to give the plates, take it out. Time was limited. Lunch time was going on. Within 20 minutes, we have to complete. So many factors were there. So there may be a chance that it may got the contaminations. Yes, this is the call the lawn growth. My God. This one is a fully a lawn growth. બધે જ ભરાઈ ગયો છે. Okay. Though it is a bacterial suspension, and if you are looking for some basic characters, elevations, textures, pigmentations, you will find so many things all together. It is like that you have inoculated with a soil sample. Okay, so it is a mixture of it. There is a lots of things. Yeah. Yes, here the yellow pigmentations, red, green, as well as the some black spots are there. If you are going to find and just check it out from the outer part, don't open, just keep on the tilted position. ઉપરથી જોશો તો some are the raised. Okay, પર્વતમાળા હશે. Okay, it's like a mountain. Okay, so many plates, different plates are there. Just observe it. Some are the green or the here. If you are looking, sometimes you will find that in one colony may be growing properly, another is not allowing to maybe grow. It may be there is a chances that this bacteria may release something. It does not allow the growth of other bacteria. Just try to observe. Several plates are there. So good initiative. Lots of things is there. There is nothing. There is something. Yes, very good. Yellow and red pigmentation. So need to handle carefully also every time. So same on a Thursday, we'll going for the like discard. How to discard it? We'll go for the media preparation for the next afterwards. So on a Thursday, we'll discard it. So that is a basic protocol even. How to handle, how to discard. Currently, we are knowing that it may be a fungi. Today is a Tuesday. On a Thursday, when we are going to open it, the bacterial growth for the fungi maybe has overgrown. Okay, maybe has completely area maybe started to get covered. This one is a very good. It's a beauty even. Microbial beauty. It is a swarming growth. Earlier, मैं तुमने अलग characters बताया था ना. So it had start to grow and spread. नहीं सारू खराब कोई वस्तु नो नसी. Any characteristics. We just looking for the characteristic. How the organism are growing. Some are here. You can see it is swarm growth. Okay, there is a growth in continuous pattern. You may look for the texture. Their outer boundaries, right? Outer boundaries are not round. It is very much fuzzy. Some are in the rhizoid. Here you can see it is like a rhizoid. The growing size, it is like a rhizoid. So just check other different plates. You may just share with one to another. So it is. હશે હશે આ ઢગલો અહીંયા પડ્યો ને બીજા ઢગલામાં છે. Let me see whether it is a fungi. આ fungi ઓલું bacteria. Yes. થઈ શકે. હા એટલે આ લોકો એને ઇન્ટ્રોડ્યુસ નથી થવા દેતા. એવું જ થાય ને. એની હા અહીંયા છે તો આ એને આગળ નથી વધવા દેતા. બરોબર છે. અહીંયા ગ્રોન છે જો આ વચ્ચેનું છે ને આ સાઈડ નું બીજું ઈ ગોળ રાઉન્ડ બનવા જાય ઓલું નથી થવા દેતું. એટલે ઓલું અઘરું છે આના કરતા. હોય ને ઘણા એના ઓરા એવા હોય ને કે બાજુ બેહવાનું મન જ ન થાય, પહેલેથી છેટા રે એને છેટા હારા. Yes, it's pigmentation, bacteria. Red pigmentation. Yellow પણ pigmentation. White? શું? હા, બેક્ટેરિયલ જ છે. યસ, જો આમાં હાર્ટ શેપ જેવું કંઈક દેખાય છે, ઇટ્સ ઓલસો બેક્ટેરિયલ. અલગ જ જાતનો, ધીસ આર ધ ટેક્સચર્સ. નહીં એ જ કે કન્ટામિનેશન હવામાંથી બધું આવે. એટલે આપણે જ્યારે અઠવાડિયા પછી જોઈએ ત્યારે ખબર પડે ને કે આપણે કેવું સરસ કામ કરીએ. નહીં, યલો નથી. જો આપણા કલરા કલ્ચર્સ નું છે ને એકેયમાં હરખું દેખાણો જ નથી. આઈધર વસ્તુ એવી થઈ છે કે એ હજી વોર્મ પ્રોપર નહોતું, ફ્રીજમાંથી લઈ આવ્યા ને એટલે બરાબર છે, એક્ટિવ એ હતા જ નહીં. અને આપણે સીધું ટાઈમની લિમિટેશન્સના લીધે ગ્રોન કરવાનું સ્ટાર્ટ કરી દીધું. એટલે ઈ આમાં ક્યાંય હજી. અને બીજી વસ્તુ શું થાય કે તમે હોટ જ્યારે હોય ટાઈમ આપણે હોય નહીં, સીધા ઇન્ટ્રોડ્યુસ કરો એટલે એરિયા મારી નાખે. એટલે મરેલા બેક્ટેરિયા બિચારા એક તો એક્ટિવ નહોતા, મારી નાખ્યા, એટલે ડેડ બોડી ખાલી ફેરવી દીધા એમ. એટલે એનું કઈ ન થયું. ના, નથી. આપણા વાળા બેક્ટેરિયા 1-2 માં તે દિવસે મેં દેખાડ્યા હતા, ગ્રોન હતા. હવે એ બિચારા છે ને yeah, this one is fungi. Oh, this was the fungi. This one is the bacteria. Fungi will be filamentous, highly. You will observe in a way that. So happy to see your results. No problem. થાય એવું જ થાય. Generally 80% to 90% failure જ આવે પેલા practical. It was expected.
That's the one type of bacteria, but here we are planting kinds of bacteria.
Yeah, the reason is that maybe while opening it has got contaminated because our plates were good. Yes, we have prepared earlier, right? It was really good. There was nothing, any error. But the other bacteria, while opening or while doing the experimentations or handling error is there. It proves that we haven't cleaned maybe our hand properly, or while doing the experimentations, we were busy in other activities. That is why I'm showing you. After a few days, that if you are going to do same thing again and again, exams you will get the same results. No, it's also not E. coli. Not E. coli. Just the contaminations. Both are the different contaminations. That is why I told that we may go for the again doing experimentation. Required to. That is the 4 quarters. Sticking will be done. Then after the 4 spread and everything. Oh wow. That is a fungi. How does contaminate? 2 chances are there. First is this, maybe a media is not proper, but media was properly autoclaved. We have taken out. There was not any error. Okay. Second problem, maybe while opening the plates, while doing the experimentations, we were talking with each other. And third, that where we have incubated. During the incubations, they were the fungal spores and other were growing inside the incubator. So our plates got contaminated. So I think second and third possibilities are more. And that is why we have shifted. Earlier we have kept into 518 to 517, the different incubators. It is like that when you are going in market, right? Something is, or you are going in jungle, that other market, you are going in jungle, you are getting some fruits. You may say whether it will be a tasty, whether it is a safe, or it will be bitter, or maybe a toxic. We don't know, right? So we need to do experiment on it. Okay, you test it, you may say it's, yes, not good. Okay. Otherwise, you'll say, wow, it is really so small, but very sweet, or you may say that a tasty, like a sour, something is there. You will try to get the more. So we can't say. With, yeah, few can be identified, like Aspergillus niger. Keep away from the fungi. First problem, maybe it is a skin infection. Skin infection is the major. Fungi going to do a skin infections. Other than that, maybe going inside your when inhalations. Sometimes they may start to grow in our lungs. That is the second problem. Or it may infect your, like in the necks and other people also getting on the eyes, like currently mucus, even in the ears. Okay, so majoritarily, so from the outer parts or the organs are there, wherever it may touch, it may start to grow over there. Okay, especially the skin growing area. Okay, where the high humidity is there. Okay, below to the hands, even to the legs, there may be more chances of getting such contaminations. If you are not knowing, okay, then after going in washroom, okay, maybe not clean. After washroom, you may have the practice of the doing the washing. But before going in toilet, we never go for the washing. That means when you leave your lab, you need to wash. To leave the things there itself. But the such practices are not there. If it is not there, that means you have taken the fungus spore with you. Then you are rubbing here and there. It is a gifting and lifting. However, you may touch or the shake hands. So gifting them to share with them that I do have the some fungus. It is the normal. We may just after doing some microbial work, you may get noticed. Otherwise, you will doing the same practices in the day by day. That is why it is advisable to its having like a mask and others. But it is up to a limitations for us. Then જોઈ ગયું બધાને બહુ સરસ કામ કરીએ છીએ આપણે ખ્યાલ આવી ગયો યસ બહુ રાખવામાં રહી જશે તો વાંધો નહીં થોડુંક આમાં રાખી દઈએ હવે એક કામ કરજો તમે લોકો જઈ 515 માં આને મૂકી દેજો આપણે ગુરુવારે એને ડિસ્કાર્ડ કરશું હું તમને શીખવાડીશ હા ઇન્ક્યુબેટરમાં હવે નહીં મૂકીએ હવે આને ઇન્ક્યુબેટ કરવાનો ફાયદો નથી બારે ને બારે રાખશો તો પણ ઈ ગ્રો થશે ઘણું બધું આમાં બરાબર છે 515 માં પેલું ઓટોક્લેવને પડ્યું છે ને ત્યાં પાછળની સાઈડ જગ્યા છે એટલે ડિસ્કાર્ડની આઈટમ આપણે ત્યાં મૂકીએ છો વોટર બાથ જેવું પડ્યું છે ને ત્યાં બે વસ્તુ મૂકતા હોય હા આને નીચેથી હરખું પકડજો અરે રસ્તામાં બધી પ્લેટો ડ્રૂમ કતી ઢગલો થઈ જાય એક એક પ્લેટ પછી આપણે આ બધી 110 કે 120 રૂપિયા વાળી છે બ્રેકેટ ચાર્જ અમથમતો ચડાવી દે પછી એમ કહ્યું કે ફી લે ને આઈ લે ને વળી પાછું બ્રેકેટ ચાર્જ દેવાનો અમારે નીચેથી ખોક આ બસ આમાં ટ્રાન્સફર ઓછી પડી ને સોરી જોજો અહીંથી આમ આમ કરીને લહેરીને પડે એની પાછી વળી આમથી આમ જાતા હોય એટલે 515 આપણે જ્યારે એક્સપિરિયન્સ સ્ટાર્ટ કર્યો હતો ને પાછળ ઓટોક્લેવ વોટર બાથ પડ્યું જ ને So when you're looking for the selective microbes, here randomly we got the some microbes, yeah. Luckily or unluckily. When you are doing with the soil sample, last time, that is why I haven't given the soil sample. I was knowing that without soil sample also we are going to get the so many things, right? If you have inoculated with the soil sample, so many beautiful color will be there. Purple, red, yet we have seen the red, yellow, some black, right? Some orange, few 4 or 5 colors is there. If you are going with the microbial culture for the soil sample, then you will get the some pink color, some reddish green, so many colors, violet, blue color will be there. So color as well as their texture, all the characteristics you can observe over it. Okay. So it is the selections. It is based on just on the plate. right? But when you are looking for this, so we are going to have the three major other aspects are there: physical, chemical, and biological. The first method is physical methods. Physical method, that is endospore formation. Maybe you are knowing for the spore, endospore. In the last semester, maybe you have learned about the spore taining. Endospore, yeah, dormant state. If you are looking for the some cells, those who are able to create a sporulent spore-like structure, so we need to give the heat treatment. Okay, how to do a heat treatment? Either on a plate itself. Okay, that is in a culture if you are going to do, or you are going to do on a staining. If you are doing, then you are doing on a slide. You may have taken the thick loop school. 4 or 5 times you have taken the loop, then you have done the heating. Okay, then you may have done the malachite green filled with the filter papers and everything. You are heating like anything. Red hot plates will be, that slide maybe has happened. So it's the heat treatment was given. So all the vegetative cell growing were there, they are going to get died, and the bacteria are pushed to compulsorily, they have to maintain their growth or their sustainability. They were going to convert it into endospore formulation. Okay, so first method for the selection of the method is like a physical method is a heat treatment. Another, it is like a incubation temperature, like psychrophiles. Those who are growing at a lower temperature, 0 to 5. Mesophiles are growing into normal room temperature to till to 40. If you're going to 40 to 45, then the thermophiles, like 60 up to. If going 60, 70 up, that is hyperthermophiles. They can grow at 100 degree even. So physical method by which we can isolate the different kind of bacteria, they are the like with temperature. So initially, we have kept our plates into incubator at 25 degrees centigrade, where the fungi were growing. So they got the opportunity. Okay, the spores are there on air. Okay, that is why it's also not continuously advisable to open and close the incubator. Everything was came from the incubator itself. Okay, fungi were growing over there. Maybe some are the useful, some are the harmful, like Aspergillus niger. Okay, it may cause to some skin disease, or at least skin infections can be possible. Okay, and you may not knowing where it may happen. Generally, yeah, be careful how we are washing our hand. We are washing with here and there. We never know the ages. We are not caring for it, especially in the hands as well as legs. In the legs, between the each fingers, if you are going to get any infections over there, it is really difficult to control. Those who are in a sports may be knowing. Is there going in a sports more? Huh? Okay. If we are those who are in sports person and playing in the ground, they are mostly going to get the infections and their age. And they are knowing that how it is difficult to control. Once it is been started, it start to grow over the places inside. So we never notice how to wash and clean, right? So these are the ages. There is a either bacterial or the fungus for mystic, and we are never much giving the attention. Well, after washing and cleaning also, right? You take the handkerchiefs. Okay, where I'm moving, my hands upper and lower. Places are there between the fingers. Never been thought, never been cared, where the opportunity for our microbes to grow will be very high. Okay, so these are some basic points are there we have to take care. So here we are looking for what? Physical method. Okay, the physical method will be there. So temperature is the another parameter to isolate certain bacteria. Okay. Then another is a pH. Some are the acidophilic lovers. They will grow at a lower pH. Major are the growing at a neutral pH between 6 to 8 or like in a 9. As you are going in above the 9, the alkaline conditions will more. those bacteria can grow in the alkaline conditions, will not grow in acidic condition. So how to prepare such medium also we have to learn. Okay, nutrient growth when we are preparing, so we have to adjust their pH. So while adjusting the pH, another problem may start. Maybe you are doing certain experiment in chemistry, that in acidic conditions something will start to precipitate. If you have prepared the nutrient media in broth, and if you have added some acidic conditions, how we can create the pH balance? pH, how you are maintaining for the any buffers? pH can manage karo? Okay, by adding the acid or base. So which acid you are using? HCl. Okay. Any other? NaOH. Okay. HCl, NaOH. For the basic, here it is acidic. Either we are going for HCl, sometimes the sulfuric acid. General, there are the several other also. Just I'm giving few examples. Okay. So when the HCl is being added, H and Cl, highly reactive species, it may start to react with one to another. They may start to precipitate the things. So while preparing the media, you will say that something is already precipitated. So we have to go for the synthetic media preparations. We need to add the different ingredients one by one. We cannot go directly the nutrient broths. Okay. Then we have to give the supplements like micro and macronutrients, the importance of micro and macronutrients to learn into it. Maybe, yes, when we are having the diarrhea, okay, or the hemophilic condition is there, people are being given with the electrolyte powder. Okay. Dust powder for the child. Why it is been given? Or even during the conditions like some person is not able to having the continuous vomiting. It's a problem, right? So over that time, generally N saline is given. Glucose bottle within the veins or the N saline is given, right? Within that, they will give the injections. Yellow or pink color solution is been added. Okay, it is nothing but the macro and micronutrients. Electrolyte is given because we need the sodium and potassium. Okay, so we know that like HCl is been added, something is been added, it is going to have the reaction with one to another. So there may be a chance that a salt precipitations may happen. Sodium it is very high in a concentration, then NaCl will going to precipitate. When you are going to HCl, the Cl with the sodium is going to bind, and we know that acid and basic reaction is always result in the salt precipitation. Chemistry, basic rule of chemistry, when the acid and base are added, what going to form? Salt, right? And the salt is essential. Without salt, if it is not in dissolved form, we cannot survive as well as the bacteria cannot survive. So now how to dissolve the salts? If you are going to rot and organic, it cannot because of pH condition. So step by step, we have to prepare the synthetic media that a pH should be get maintained. They are the salts. What are they? Are going to remain as such. Then we need to go for the different combinations. Like if you are going for the calcium carbonate, we need to go for the others while mixing it. We cannot go for the natural sources. So that part we have to learn. Right now we are not going to bother about it. Just I'm giving the few examples that acidophiles we can create. We can go to the basophiles or the alkalophilic organisms we are telling that can be grown over it. But it having their own limitations. Media preparations are going to change according to our sample. Okay. So some example is given lactobacilli from the like buttermilk present in the cheese or the butter. pH of the medium is maintained 5.35. Okay, high pH medium selection for the alkali tolerant bacteria. Those can tolerate the alkaline condition. So we may go for the alkaline conditions will be developed. Some are the medium pH like Vibrio cholerae. What is the Vibrio cholerae? What it causes? Cholera, right? That's cause cholera. Okay. Then we may go for their isolation, slight alkaline condition. Not a basic also, slight alkaline conditions we are growing. Physical method, cell size and mobility. Like other example, Sometimes we are looking for the motile and non-motile cells are also there. Here I'm not sure if it is there or not. One method is called the hanging drop method. Hanging drop method is called. Into it, we can check the motile cell. Motility of the cell can be observed under the microscope. Have you done that experiment? Hanging drop? No. Okay. I just forgot to bring the marker. No problem. Just I can give the few examples. Generally, you have seen the slides. Glass slides are there. Okay. What is their surface? It is flat on the both the place. While in case of a cavity, slides are specifically are available. It's like a watch glass. Slide is there only. Within the middle of the part is somewhat thick. Over the middle part, there is a, you can say, one notch is there. It's like a round bowl-like structure is there. So what we can do, we may put our sample. Like it is like a nutritive from the sample, soil sample you have prepared, or the nutrient agar or the suspension is there. It at least requires the suspension for in liquid form. That you need to put the drop over it, okay, and cover with the cover slip. Okay, put with the cover slip. So your drop is there within it. I'm going to put the cover slip. Okay, and what we are going to do, we in the their cover slip, it's covered with some Vaseline. Okay, we are going to stick with the Vaseline, or we may use the other materials like oily substance that can be used. So our cover slip going to stick over it. So my sample is there within it, and I have completely covered. Now invert your slide. So your drop is there over here. Okay, drop is there where in the middle of the in the cavity. Now put that in a microscope slide. Okay, only motile cell you will observe they are swimming. Start to swim within it. So some particular organism are having the high motility. Some are very low. So you can very easily observe. This is called the hanging drop. Drop is hanging on the cover slip. Hanging drop method to check the motility of the cell. Okay, so here also cell size and motility. Yes, majorly motility is caused by it, but some are having the pilia-like structure. Small tentacles like part is there. Flagella is the major. Maybe you have learned the flagella structures different. Mono, tricus, dicus, many, polytricus, flagella. Okay, so they are the for the motility. So we have to just check it out whether it's having the such or part or not. Okay. Even here, some basic experimentations related to hanging drop related is sperm collection. Okay, that is from the human or from the animal is also there. Okay, so when you are looking for checking, those who are in path lab, those who are working in the path lab, in the sperm banks are there, or those who are in the IVF related activities are going on. That the major first checking is done related to the sperm motility. Okay, when people are going in IVF, in vitro fertilizations, either one either male or female having the some problem. Okay, they are not going to get the babies. Some problems may be there, so we have to check from that. So male is diagnosed differently, female is diagnosed differently. Okay, so motility is one of the important factors. The sperm ejaculation is done, but they are remain there itself. Their swimming and capacity for the move in the liquid may be lesser. Okay, then the medicines are described into it. So such method for the hanging drop, if you are looking, then the sperms are moving very fast if it is the active state. If it is not, then it is non-motile. Okay, yeah, if you are doing with the sperm, the collection of the sperm and observations, that window, it's called the window period, observation, should not be a prolonged. Okay, it should not be a prolonged time. If the sample is collected in the morning and if you are checking at the night, the whole cells is on the dead condition. Or maybe if you are live, otherwise in dead conditions. That is a time period, window period is called, it should be a minimum. Okay, then you can observe the cell motility. Right, it can be a possible into it. And yes, cell size. that is like membrane-like filters of the size can be placed on agar, and the oral scrapings are placed on the filter. Generally, so many methods are also there. We may have the some filtrations also. If you are looking for the some Millipore is a company is preparing the certain filters. So if you pass the some suspension from it, small organism will pass, other will going to. It's like a sieving. So based on that also you can measure. Otherwise we may go for the like a electron microscopy. To measure the cell size, we may go for the electron microscopy. You can observe the till the there what are the organelles could be possible within the organism. Okay, what are the organelles present in the bacterial cell? That's I'm asking. In bacteria, which organelles will be present? Nucleoid. Nucleoid. Okay. Organelles present in a bacteria. Yeah. Do you know any organelles? They are present in bacteria. Cytoplasm. Cytoplasm is organelle. Plastid.
Some are like
Cyanobacteria having plastids. Okay. Huh? Yes. Ribosome will be there. Good. RNA. RNA is the organelle. How? Golgi apparatus will be there in bacteria. From where Golgi apparatus is going to arise then? The basic difference, is endoplasmic reticulum will be there. Ribosome માંથી પણ. Okay, ribosome હોય. Golgi apparatus હશે? Okay, from where the endoplasmic reticulum arise? No થાય રે. પાછા આવી જાવ. Nucleus માંથી હોય. Nucleus હશે? Prokaryotes do have the nucleus. You are learning the biology, microbiology now. Okay, then you are learning the prokaryotes. Okay, you are asked which for the bacteria. The cell biology basic things. You don't know who are they. What sort of the, you can say, kind of a dragon. You don't know, right? And you are learning them. Tomorrow or whenever my next class will be there, come with the difference between prokaryotes and eukaryotes. Okay, basic structure of the bacteria you need to show to me. Okay. I'm asking for the many things, right? Basic structure of the ultrastructure of bacteria. Come with the learn ultrastructure of bacteria, and another is a difference between prokaryotes and eukaryotes. I don't know. You have already learned. I don't know now. That was a 10 plus 2 and the plus 1. Okay, already you have came in the 12 plus 2 now. So there I don't know. What are the organelles present? Difference between prokaryotes and eukaryotes, and another is the ultrastructure of bacteria. Come with the drawn and the written part. Okay. Otherwise, what I am telling or discussing, it will be of a no use. આપણને ખબર જ નથી કે શું ભણી રહ્યા છે, કેમ બેક્ટેરિયા વિશે જ નથી ખબર. મોટી મોટી વાતો તો ગણી કે નાખી. Yes, another. What are the things are included? We are looking for isolating them, right? Physical method, chemical, so many things. But to whom we are isolating, that we are not knowing, right? Okay, what are being included into microbes? Virus is a microbe or not? No? Yes? Yeah? Okay. Another question. Bacteria is a microbes or not? Yes. Fungi is microbe or not? Okay. Algae is microbe or not? Huh? Yeah. Okay. Protozoan amoeba is microbe or not? Yes. So whether it is of all are, and the algae, protozoa, fungi, bacteria, okay, and viruses I'm asking, all are prokaryotes or they are eukaryotes? Okay, some are microorganism. So what is the definition of microorganism then? We can see. We can't see. Okay, can we see the algae? Oh my God, then is a confusion. Okay, some are macros, so they will be microbes. So what is the definition of microbes then?
Organism with size in micro.
How many that is? Micrometer. Ah, okay, okay. I can measure this thing in micrometer. I can convert with the unit. Is it possible? I know it. Same thing. If it is, I'm telling the 10-inch. I can convert into meter, can convert into kilometer, can be converted into millimeter, micrometer. That is just unit. So what is the definition? What is the definition of microbes then? So do the virus is included? Do the swarm is included? Now I'm telling that the fungi, already you are knowing that is microbes. Amoeba, it's the largest size. Whether it is the microbes. So what we are learning? Microbiology? We don't know the what to learn then. I will ask such question in Viva, right? Same thing will be repeated. Basic things I will ask. Nothing will be there in my special class. Okay. In Viva, I will ask many things related to that only. You will say that, sir, what about the papers? Okay. So you are knowing basic things or not, doing the brush up related to your subjects or not, will be shown or reflected over there. So you don't know the microbiology, don't know the microbes. You know, so come tomorrow somewhat prepared. Definitions for the microbiology subject. What we are learning in microbiology? Microorganism. Okay. So what are the microorganisms? Okay. Okay. We can't see. So we can see the amoeba or we can't see the amoeba, fine, but we can see the fungi. Mycelium will be there. Yes, very easily. It is a mucor. It is a microorganism, or it's a tiny molecule or the larger organism. Even though we are calling it a microorganism.
See, like we are doing the culture of bacteria. Still, then we are able to see those features. That is the colony.
Okay.
Colony which start growing. Then we can study the micro.
Yeah. Good try to convince me, but it is yet not fully.
You can see a single.
Yeah. In a colony. Okay. I may take the skin cell, start to grow on a plate, observing on a microscope. Is it a microorganism? Is it a microorganism? It start to grow in a colony. Animal tissue culture, you are going to learn, even in plant tissue culture. Okay, if you just take some leaflets, put on the media, it will start to first convert it into callus. Do is called the microorganism. Because to study, to observe, to learn about them, we need the can't see with naked eye, so we need to use the microscope. So it will be converted into the microorganism. So plant is a microorganism now or not? Mucor, we can see mucor. It's a fungi with naked eye. You can observe it's mucor. Okay. So what is the what is actuality? What you are learning there? I'm telling the fungi we have received many things, right? Which organism? I will come one by one. Yeah. Okay. So what is the difference at the cell level? That's why I'm asking. What will be the difference at the level of the cell? No. About okay, cell difference in cell. Yes. So this I'm asking by the prokaryote and the eukaryotic difference, cell difference. But then you will say that some microbes are coming here in eukaryotes and prokaryotes. Then the next day questions will come arise again. Yes, madam. हाँ अच्छा મ્યુકર હાલો એને નથી ગણતા મ્યુકર આવડું મોટું હોય બિલાડીનો ટોપ જોયો છે અચ્છા so if I am taking the skin cells તો કઈ આવે ને કઈ ના આવે ઓકે give the reference કે આવી રીતના માઇક્રોબાયોલોજી માં કહેવામાં છે આ ફંજાઈ ને આપણે માઇક્રોપેસુના ને નહીં કહી give the reference if you are telling that is you trying to convince but it is not come up with the answer then come with some proof or the reference go study microbiology by the Pulsar. One book will be there, first, second, third chapter will be related to it. If you are not coming with a prepared mind, if you are just learning, you can learn. But after completing the years, you will say that I'm normal. You don't know the basic concept. Basic difference you are not knowing. You are a so-called the microbiologist, and if you are not able to define the micro, fungi, other things, it is of no use. You are not knowing the reality and you are learning that. Right? If you are learning the science, everyone maybe have taken the after 10 plus 2, maybe here in the outside country, all the science as a subject. At least one as a science as their subject, and it has been asked, what is science? And the another difference I'm asking the technology. Difference between science, technology, and engineering. Can you differentiate three terms? Science, technology, and engineering. You are coming in the faculty of science. What's the difference in the faculty of engineering? What's the faculty of technology?
Biology, science, and technology is made by humans.
Okay. Science is not done by human. Okay, technology is done. Okay, what is then engineering? Made by robots. Robots is been prepared. Okay. They don't do research. They will prepare the structures, anything. No research. Huh?
The knowledge, everything is science.
Not everything is science.
The knowledge is study of science, but application becomes knowledge.
Okay, that is science.
That's technology.
It's technology. Application is technology.
Yeah.
Learning is science. So what is engineering then? Somewhat near to, somewhat you have touched. So એને શું કહેવાનું? માઇક્રો મેક્રો આલ્ગી મેક્રો મેક્રોફંજ ઓકે માઇક્રો ઓર્ગેનિઝમ નથી નથી લિટલ બિટ સો ई બે માં શું ફેર પડશે મેટાબોલિઝમ માં કઈ ફેર પડી જશે નાના મોટા થઈ ગયા એટલે ઓકે ઓલું પ્રોકેરિયોટ છે પાછું આ લે કા ઓકે યસ અડધા યુકેરિયોટ બીજા બધા I really don't know. I came to you people expecting that at 9:30 when closing my lecture that you are knowing the difference between the prokaryotes, eukaryotes, difference between the microbes, what are they included in microbiology, because you have completed, passed out your 10 plus 2, then you have passed your first year even. So I'm expecting that you are knowing, and that's why I came to learn from you. Then I can tell more. Okay, so I hope just stopping here, put your attendance in the attendance link. So come tomorrow with learn at something. Yes, and if whatever you are telling, give them some references. If you are knowing, okay, it's not like a Wikipedia. When you are opening, first is coming the Wikipedia. Wikipedia ma બધું editing થાય ખબર છે? You can also edit. You can just log in. Okay, you just log in the Wikipedia. You can also edit it everything, but they are continuously monitoring. If you are going to do falsified things, you will be blocked. If you are using the Gmail, that email ID for the Wikipedia will be blocked. Okay, so don't just rely on the Wikipedia. So I'm ending here. We will meet in the next lecture.